thanks to the organizers for inviting me. It has been very exciting to have a different perspective than the one that we usually have in the lab. And so today I'm going to talk about one strategy that we have started to use to try to deconvolute very complex transcriptional networks in Arabidopsis. And I will try to focus on or give you a sort of historical perspective on how this strategy was evolving in our hands. So to give you a little bit of background, as you probably know, we live on a planet that rotates on its own axis. And this, of course, generates periodic environmental changes. The most obvious ones are lidar cycles or temperature cycles that occur every day. And probably for this reason, most organisms that live on the planet have evolved to perform many biological functions in a time of the day specific manner. Not only this, but most of these organisms have evolved an endogenous oscillator, or known as biological clock, that actually drive these circadian rhythms in many different processes. Okay? And in the case of mammals, the most common example is the regulation of sleep-awake cycles, or regulation of glucose or cholesterol biosynthesis, and so on. And in plants, as Sam sort of introduced, the clock regulates, at least in Arabidopsis, almost every aspect of plant growth and development. So we can see that there are many biological functions related to photosynthesis, hormone production, leaf movement, flowering, nutrient uptake, sensitivity to hormones, and so on, that actually occur or are more important at specific times of the day. And for the most part, we know that, as it was well mentioned in the introduction, that about 1/3 of the whole Arabidopsis transcriptome is regulated by this mechanism. So it's very common to find that genes that belong to different, or that are important for different pathways, are actually regulated by the circadian clock. So, and of course, we use gene expression as a readout to analyze the function of the circadian clock in plants and in other organisms. And one strategy that is widely used is to generate transgenic organisms. We generate transgenic plants that carry a luciferase reporter gene whose expression is driven by a clock control promoter. So now those plants are going to produce luciferase in a rhythmic way. And of course, photons emitted from that plant are going to also follow a waveform that is repeated every day. So in a typical experiment, what we would do is to grow our organisms in lidar cycles and then transfer them to constant light conditions. In constant light conditions, we can actually analyze the function of the circadian clock isolated from mechanisms that are directly regulated, for example, by the lidar cycles or by temperature cycles. And then what we do is to image these plants at regular intervals for at least 4 to 5 days. And after that time, what we obtain is a set of images that build a movie more or less like this. So you can see how these seedlings are glowing rhythmically. So what we can do after the experiment is done is to actually quantify the photons coming from each plant. And we can plot the information for each seedling, so you can see how the expression of luciferase, the luciferase activity, changes every day. And we can actually use these plots to quantify some parameters, or to obtain some parameters that allow us to quantify the function of the circadian clock. The most commonly used one is the period length, which is essentially the time lapse in between one full cycle. Okay? As you can imagine, for wild-type plants, this period length is usually 24 hours. So when researchers, started trying to understand which were the molecular mechanisms that underlie the function of the circadian clock, what they did was to follow classic forward genetic screens, where they would use these organisms to mutagenize them and look for phenotypes, okay, that look like the clock function was altered. Of course, the most obvious one is arrhythmic phenotypes, but also they were found short period phenotypes or long period phenotypes. Okay? And then what we can do, or what they did, was to map these mutations, identify the components that were mutated, and then try to start establishing the links, molecular links between the different components. And what they found is that actually, or they came up to the basic molecular architecture of the circadian clock in Arabidopsis. And this architecture, as in other organisms, is built on multiple interlocked feedback regulatory loops. So now one interesting characteristic of these feedback regulatory loops in Arabidopsis is that for the most part, they are built on the transcriptional regulation among the different clock components. So usually, one clock component is regulating the expression of its own regulator. Okay? Although most of these relationships were established based on the transcriptional regulation among the components, only these two genes, at least when I started my postdoc, were canonical transcription factors, CC1 and LHY, that were in the network. The rest of the genes, or the rest of the proteins, did not have any canonical DNA binding domain, and therefore it was not certain how these other components could feed back on the regulation of their targets. So one of the things that I wanted to do when I started my postdoc was to try to fit this network with transcription factors. So certainly transcription factors are part of this network, and that are actually explaining how many of these genes are regulated. So together with Sam Hazen, when I just started, we continued doing forward genetic screens, and we used a combination of a rapid array mapping using DNA microarrays and also PCR mapping. And we mapped about 60 mutants, 60 clock mutants. What we found is actually that all those mutations were actually in already known clock components. We identified a number of new alleles of already known clock components. So this was indicating us that very likely these forward genetic screens were reaching saturation, or at least in the way we're doing them, they were reaching saturation. And this could be essentially because of the redundant nature of the network, and also because, and thinking about transcription factors, we know that transcription factors in Arabidopsis and in plants in general are usually have duplicated, and they have very redundant functions. So we thought that probably this was the reason why we could not identify transcription factors associated to the clock network. So what we decided to do is to go backwards, to try to establish a reverse genetic screen to try to first identify transcription factors that are directly interacting with the promoters of clock genes, and then try to figure out whether those transcription factors are biologically relevant regarding the function of the circadian clock. So to this end, what we decided to use is a very well-known method to follow or to try to search for DNA protein interactions. And this is a yeast-1 hybrid system. In the yeast-1 hybrid system, we have two basic components. One is a yeast reporter strain, where a reporter gene is expressed, driven by any promoter region that we are interested in studying. And we are going to transform this yeast strain with plasmids that encode transcription factors that potentially bind to the promoter region. And of course, the outcome is pretty obvious. If the transcription factor, based on its own intrinsic DNA binding capacity, can recognize the promoter region that we're using as bait, this transcription factor, I forgot to mention, that is fused to a strong activation domain, so now the expression of the reporter is going to be enhanced, induced. And that colony is going to have an increased expression, in this case, of beta-galactosidase activity. Of course, if the transcription factor does not bind to the promoter that we're using as bait, we are not going to get any beta-galactosidase activity. Now, one of the problems that the one hybrid system, at least performing in the classic way, one of the problems that it has is that originally people started using libraries that were constructed by extracting total RNA. And usually in those preparations, transcription factors are not very abundant. So because we knew the Arabidopsis genome had been sequenced, what we decided to do was to actually try to perform a gene-centered approach, or try to establish a gene-centered approach where we could first clone every single Arabidopsis transcription factor, and we in a vector, of course, suitable for or compatible with these 1 hybrid screens. And then we would organize those transcription factors in 96 well plates, as we originally thought about it, and such that in each well of this 96 well plate, we can test the interaction between our promoter bait and one single transcription factor. So in each well, we are testing one single pairwise combination between transcription factor and promoter bait. So as a way to validate this system, and because there are so many transcription factors encoded in the Arabidopsis genome, what we decided to do was to actually create a small collection of transcription factors. We created a collection of 200 transcription factors that are actually regulated. The expression of these transcription factors is actually regulated by the circadian clock. So the rationale behind this is that most clock components are themselves, the expression of most clock components is regulated by the circadian clock. So if we were able to, if we wanted to identify transcription factors that are part of the clock network, very likely these transcription factors would be regulated by the circadian clock. So then what we did was to look at the promoter of a key clock component that is called CC1, as I just mentioned, and then we combine different promoter fragments of CC1. We actually screen individually different fragments of the CC1 promoter against the whole collection. So this is just a typical result where you have, most of them are negative results, but you have a few wells where the beta-galactosidase has increased. So because you know what's the transcription factor that is present in that particular well, now immediately you know the identity of the transcription factor that is interacting with the promoter fragment that you're using as bait. So there is something wrong here. Okay. So the figure has changed. So basically, by performing this screen, and I'm going to, unfortunately, the figures have changed. So by doing this screen, what we found is actually one transcription factor that, as you can see, interacts specifically with this fragment of the CC1 promoter. This was a transcription factor that belongs to the TCP family and that we decided to call Che. And one of the things that we envisioned when we started doing these screens was that plant promoters were going to be too long to actually be fully functional in yeast. So this is why we decided to actually use shorter fragments of the, encompassing the whole promoter region to actually perform the screens. So for example, for the CC1 promoter here, we actually performed the screen with 5 different promoter fragments, okay? And the proof that this is true is when we did the experiment using the full-length promoter. So when we did the experiment with the full-length CC1 promoter, we were not able to detect the interaction of this transcription factor, okay? Really implying that by using shorter promoter fragments that are probably more functional in the yeast system, now we can actually detect transcription factors that bind more to the distal portion of the promoter, of the plant promoter. So, of course, once we identify transcription factors in a yeast system, what we need to know, what we need to look for or try to analyze is whether this transcription factor is actually biologically relevant for the plant, okay? And in particular, in our case, for the function of the circadian clock. So what we did was to overexpress Che in Arabidopsis plants that carry a CC1 promoter driving the expression of luciferase. And as you can see here, while the wild type traces are in black, and 2 overexpression lines are in blue, and you can see how the overall amplitude of the oscillations is reduced in the overexpression lines, and actually the phase of the peak is different. The peak in the overexpressors is about 1 to 2 hours earlier than the peak of expression in the wild-type plants. So these and other experiments that we performed indicated that actually this transcription factor Che is relevant, is biologically relevant for the function of the circadian clock. And actually we performed chromatin immunoprecipitation assays that show that this transcription factor actually binds to the promoter of CC1. So these and other experiments allowed us to actually establish a novel feedback regulatory loop where Che, this TCP transcription factor, directly regulates the expression of CC1, and in turn, CC1 can regulate the expression of Che. So again, this follows the general trend of the architecture of the circadian clock, where we have these interlocked feedback regulatory loops. Now, most important is that this work actually validated the use of this transcription factor-centered collection to actually screen for transcription factors that bind to essentially any Arabidopsis promoter. So the only thing that we were missing to actually extrapolate this strategy to any Arabidopsis gene was that the collection has to include all the transcription factors that are encoded in the genome. So that's exactly what we tried to do. We actually tried to generate a collection encompassing all the transcription factors that have been predicted to be encoded in the genome. Okay? And to this end, what we did was to actually look for genes that were called as transcription factors by many different laboratories, and we looked at different databases, and we compiled a set of about 2,500 genes that encode for transcription factors. And in this graph, I'm just showing transcription factors that are, or genes that are called as transcription factors by the 4 databases that we included in our analysis, or 3 databases, and so on. And then we went through, we didn't have gene synthesis, so we went through a very thorough and reiterative, I would say, process of amplification of each gene, cloning into a vector backbone, and then sequencing each clone to make sure that the sequence was accurate. And if the sequence was accurate, we could keep that clone for our final collection. If it was not, then we would start the process again. And we repeated this process on average 6 times for each gene that didn't work that didn't work. And what we obtained at the end is about 2,000 clones, 2,000 transcription factors that here, I'm just showing in green the transcription factors that we cloned for each one of the categories that I'm showing over here. And these 2,000 transcription factors represent about 80%, 78.5% of all the transcription factors that are predicted to be encoded in Arabidopsis genome. Now, if we look at the transcription factors that we have cloned, we find that for the most part, those genes, the genes that are present in the collection, have a sequence that is exactly the same as the sequence that has been annotated. We included a few genes, 90 genes, that likely acquired a silent point mutation probably during the amplification process. And we actually included 19 new gene isoforms that are genes that consistently, in every single round of amplification and sequencing and so on, gave exactly the same difference comparing it to the annotated gene. So we decided that those were likely to be novel gene isoforms, and for the most part, we find that in these 19 genes, in most of them, we find that there is a partial retention of 1 or 2 or more introns. And of course, we will also analyze, what happened with the ones that we couldn't amplify. And I'm not going to go into details here, but this essentially is allowing us now to try to establish different or alternative cloning strategies to try to actually expand the collection to include as many transcription factors as we possibly can. Now, one interesting thing is that if we look at the transcription factors that are present in this collection, and we look at how many of them are represented in microarray data sets, we find that about 20% of the transcription factors that are in the collection are not in microarrays. So now, by having them in the collection, we can probably start assigning a function to these transcription factors that when we analyze large microarray data sets to look for correlations, for example, we would be missing. And then the other important thing is that the whole collection was built in a vector backbone that is compatible with a gateway recombination system or cloning system. So we have a full collection where every transcription factor coding sequence is cloned in a gateway donor clone. And now what we can do is to transfer this coding sequence to essentially any destination vector that we're interested in. So in our case, we transfer it to a yeast expression vector that allows us to do yeast-1 hybrid screens. But you can think of transferring these transcription factors, for example, to a bacterial plasmid to express these proteins in bacteria, or to a binary vector to actually generate plants that overexpress each one of these transcription factors. And again, here we did 3 rounds of recombination reactions to actually obtain a full sister or daughter collection, I would say, of clones that are suitable for yeast-1 hybrid screens. So, of course, expanding the collection brings an additional problem, which is a number of technical problems to actually process all the plates that we need to process to screen each promoter. Remember that for each promoter fragment, we are, on average, screening 3 to 4 shorter promoter fragments. So this multiplied by 2,000 transcription factors, it means that for one average promoter, we need to actually screen or analyze about 8,000 wells in, to actually be able to analyze transcription factor promoter interactions individually. So we actually were able to set up a robotic platform and to actually optimize the whole screening system so we can now perform all the steps in an automated fashion. I mean, one of the improvements, of course, was to move our collection to 384 well plates. And then I would say that overall, the main thing that we worked on was to try to make every single step in a liquid format. Okay? And one of the main improvements that we have introduced to the system is the use of a novel reporter system where we actually express the reporter protein, in this case it's a luciferase, associated to the membrane of the yeast cell. So that now the luciferase is facing to the extracellular compartment. And this really was created a significant advance in terms of the manipulation of the plates and also in terms of how long the whole process was taking us. And essentially this is because for the most part, most of the reporter genes or most of the reporters that are used in yeast require, when you want to actually measure the activity of the reporter, you need to actually physically disrupt the membrane of and the cell wall of the yeast to actually allow the substrates to go inside the yeast and to actually be able to measure the activity of the reporter. So by using this system, now we can actually measure the activity of the reporter in just a crude cell extract, but in a crude growth, cell growth. So with this system, we can now actually screen about 30 to 40 promoter fragments in about 1 week. So this represents about 80,000 transcription factor promoter interactions that we can check individually in around 1 week. So one of the advantages of actually measuring the activity of the reporter is that then for any given promoter fragment, we can actually plot, okay, what's the level of activation that we observe for the reporter gene. And this allows us to actually categorize the interactors, okay, based on how much they induce the expression of the reporter. And this allows us to actually prioritize which are the transcription factors that we are going to follow up in, to actually establish if they have a biological relevance in plants. So if we use this larger collection and we screen again the different promoter fragments that we had for the CC1 promoter, what we obtain is that about 300 transcription factors. And this is, I would say that the CC1 promoter is unusually sticky, I would say. For most of the promoters that we have screened, we get on average about 50 interactions. In this particular case, in the different rounds of screening that we have performed, we usually get more transcription factors than for the rest of the genes. But what I wanted to point out here, these different squares of different colors are representing the different short fragments that correspond to the CC1 promoter that we use. And as you can see, most of them actually are bound by a relatively similar number of transcription factors. The only one that has a completely different number is this promoter fragment C3 that we know it has a very high level of autoactivation of the reporter. So in the reporter strain that carries this promoter fragment, we actually have a very high level of beta-galactosidase activity or luciferase activity, even in the absence of a transcription factor. So that's why now it's difficult to differentiate, a positive interactor in a strain that already has a very high background. But one interesting thing is that for the most part, all these green dots are the different transcription factors, okay? And all the ones that are in the outer circle are transcription factors that interact with only one of the 5 promoter fragments that we're using as baits. So about 72% of the transcription factors that came up as hits in these screens are interacting with only one single promoter fragment, which actually means, or to some extent, it means that these interactions seem to be highly specific, okay? only a small or a smaller percentage, about 20% of the transcription factors that we found as hits are interacting with more than one of these shorter promoter fragments. So we are actually in the process of trying to find tools that would allow us to rapidly validate these interactions. Again, these are interactions in yeast, so we are working on different methods to try to validate these interactions in planta. Okay, so we are in the process of that, but I would like to just mention briefly one example of one of the transcription factors that we find in these screens. And I think this example really represents how unbiased these type of screens are, and you're going to understand this in a minute. So this is a VHLH transcription factor. Again, it interacts with a different fragment, but only with one fragment of the subfragments that we were using, encompassing the whole CCA1 promoter. And as you can see, it binds specifically to the CCA1 promoter. And when we did the validation in planta, when we overexpress it, what we see is that actually there is a reduction, okay, and again, a slight change in phase in the oscillations of the CCA1 expression, okay, indicating that very likely, again, this transcription factor is biologically relevant for the function of the circadian clock. We were able to actually confirm that this transcription factor, by chromatin immunoprecipitations, we confirmed that this transcription factor binds to the CCA1 promoter in vivo. And most importantly, what we found is that this VHLA transcription factor that we call TCF, the expression of this transcription factor is induced by warm temperatures. So what we decided, based on this characteristic of the expression of this transcription factor, is to actually look whether misexpression of this transcription factor would have an effect on the temperature responses of the circadian clock. And what we actually found is that plants that overexpress this transcription factor exhibit a phenotype that is dependent on the temperature at which we do the experiment. So you can see how the wild-type traces here are in black, and the colored ones represent this VHLH overexpressor. And you can see how this phenotype really increases as we increase the temperature. And one of the characteristics of the circadian clock in many organisms, and I'm just finishing with this, is that circadian clocks can compensate to changes in temperature. So you can see how, although we change the temperature here, the period length of the oscillations is the same at 22 Celsius and 28 Celsius. However, in these VHLH overexpressor lines, the period length is significantly shorter compared to the wild type, indicating that this temperature compensation mechanism has been somehow disrupted by this transcription factor. So I would like to just summarize this by mentioning that, by these two examples, we have been able to validate an alternative strategy that is allowing us to actually find direct regulators of, in our case, clock-regulated genes, but we have been using it with many other laboratories to find interactors to promoters of genes that are important for many different biological functions, and some of those works are already published. So I will bypass this, and I will just finish by acknowledging my postdoctoral mentor, Steve Kay, where I did most of this work, Ghislain Breton, who was the one pioneering this work, and then key collaborators that have allowed me to finish this work now, Don Nagel and Colin Doherty. And with this, I will finish. I'm sorry if I went a little longer.
We should only have time for a few questions. Asaf.
Hi, good talk. So in fungi, for example, in Neurospora, for circadian clocks, the master transcription regulator, like frequency, which regulates other transcription factors and other genes, and then other transcription factors regulate genes. So do you see the same hierarchical nature of transcription factors in Arabidopsis?
That's the hypothesis, yes. So the hypothesis is that there are different layers of transcription factors where clock components regulate the second layer. Is that what you mean?
Yeah.
Yeah. And so, so we, in my lab in particular, we are focused on pathogen-related genes, and we are trying to use this type of strategy, among others, to try to establish the connections between clock components and the regulation of key genes that belong to the, to specific pathogen response pathways.